So I'm going to do sort of what the title of this talk says, and it's really my rethinking of autism and of animal models in particular that will be the thread of this talk. And I'm a systems neuroscientist and a systems thinker. As Alice pointed out, I try and understand how things work at all the levels of biological organization to the extent that I can. And today's lecture is going to focus really on the electrophysiological manipulations and recordings that we were able to make. But I should just say the program in general really tries to accomplish what may turn out to be the impossible, which is to understand the true complexity of the nervous system. So I'll get started. Well, I was taught to make sure that it was clear where I was going. So this image, in many ways, is a cartoon for what I want to talk about. We're going to talk about mice in their entirety. We're going to study and talk about place cells. That's the color-coded map there. These are neurons in the mouse and rat hippocampus that signal where the animal might think it is. At least that's what the signals look like. And the bars there represent a raster or set of rasters from the population activity of neurons that we'll be recording. And we're going to try to understand how the rat's behavior is reflected in these representations of place cells and in the interactions amongst the cells. And the take-home message here is really that in at least our Fragile X model mice, where we've knocked out the Fragile X mental retardation protein making gene, that the neurons in this brain look like pretty good neurons. There doesn't seem to be that much wrong with the neurons when you look at them individually, despite all of the other problems you can recognize in the model. And it's led us to believe it and to argue that actually the cognitive, at least, deficits in ASD and autism spectrum disorders, and in particular in Fragile X, may actually arise not because of the neurons themselves being poorly developed or poorly established, but in particular in how they interact with each other. And that is a really useful way to frame this problem, because we actually have a fair number of tools at our ready that can actually influence how neurons that happen to be individually pretty normal interact with each other. So hopefully you'll consider this an optimistic message by the end. So that's where I'm going. Let's talk about animal models to begin with. I should just add, please feel free to interrupt me. If something's not clear, I'll be happy to clarify it as we go. So if you look, this I got from the Spectrum website here, heralding what has been a program in ASD research and GWAS studies, which is to identify the genetic underpinnings, if you will, or the contributors to things like autism. And so at the current count, there's 100 or so genes that are in some way predisposing people to have these symptoms. So 100 is actually a relatively small number given the number of genes available. So that's in some sense good news. However, you can also find in the literature quite a number of these examples. This is my favorite one, which had 313 authors. That identified that, in fact, in these 5 different neuropsychiatric disorders, that they all shared the same genetic predispositions. So the specific alterations of the genome could end up through some program. Maybe it's a developmental program, maybe it's because of some environmental influence, who knows, but the same genetic disturbances could give you any one of these disorders, which obviously have very different symptomatic presentations. So how does one think about that in terms of trying to understand how the genes lead to these symptoms? And so that's been something that's been puzzling me for some time. And so when we have animals like mice, since we understand the genome very well of mice and Drosophila, less so rats, what we're able to do is mimic in many ways the genetic defects in these animals. And the question is, what are we really trying to accomplish when we do that? We can, if you will, lead off with the genome. We've identified particular targets that we're able to manipulate on the molecular side of things, and we actually would like to explain some of these social deficits or social peculiarities, the behavioral peculiarities, the cognitive symptoms, and there's an enormous gap in between these two levels of understanding, and it turns out that we know a fair bit. I'll use my pointer. We know quite a lot about this level and a lot about this level, but there's this enormous gulf that we're challenged if we can to bridge. I like to use this cartoon. I call it the circle of being, if you will. You can see that there are interactions between the genome and the products of the genome. For example, the proteins that might end up in membranes that actually control what a cell might or might not do. Those proteins and their location in the membrane can define synapses and how synapses might function. Those synapses themselves give rise to what you might recognize as neuronal cell types. Those cell types form circuits. Those circuits perform in conjunction with each other in different parts of the brain, and the brain obviously gives an individual their individuality and their behavior, and individuals interact together socially. And so one of the hopes, if you will, here's where that gap is. To understand that gap, you actually have to go all the way around this chain of different levels of biological organization and interaction, and that presumably is very difficult, and we're looking for the rules of understanding. And in fact, since we don't know most of the transfer functions between these different levels, we're kind of stuck in how to make progress today given our ignorance. And so that's something we think about. Importantly, this is where mice and fruit flies, the kinds of creatures where we can manipulate the genome, can actually give us some insight in, at least hope, in sort of unraveling or at least understanding how to think across these levels. And I'll give you some examples of that during the talk here. What I was able to also get from the Spectrum. In fact, most of my talk could be culled, if you will, from the Spectrum website. I hope it's well read here. So, you know, what really is the purpose of an animal model in general of something that is a brain function or dysfunction? This has been discussed, and there are a number of articles that you could read about that. The traditional answer to that is this. Usually it's thought, at least when I was starting this kind of work in the literature, these animal models are typically treated as if they're meant to somehow mimic the human condition. Okay? And if you do that, then you ask questions like this. How valid is this model as a mimic of the human condition? Does it actually are the symptoms there similar to what you might imagine? Is the etiology, the way that the symptoms arise or the manipulation arises, does that match what you might find in the human condition, and so on? It's this program in asking questions about the mimics, if you will, of animal models for human diseases, especially that of the mind, has proven to be very disappointing, to put it mildly. And so this is not how we're going to think about this. And in fact, you have to, for the rest of the talk, abandon worrying about whether the model I'll be using is or is not valid according to these criteria. There's a rather different way to think about these animal models. And the models and the way we will use the models, in fact, I think the way most scientists actually use these models in the laboratory is really to enhance, right, to engage in some sort of knowledge gathering about the human condition itself, not necessarily to mimic the human condition, but to learn something about the human condition. And as a scientist, you can characterize that really by looking at it this way. These animal models are, in particular, they're especially powerful, and I hope to convince you of that, when we use them not so much as subjects, but as reagents, right? The kinds of things you would go to a chemistry bench and grab so that you can actually evaluate or test hypotheses about what you know about the human condition. I'll give you a concrete example of that for the rest of the talk when we talk about the Fragile X condition. So we're going to use Fragile X mimicking mice, if you will, not so much because they have anything in common with the disease per se, but because they allow us to test some ideas about the disease. And I'm going to pose two distinct hypotheses that are in the literature about how the symptoms arise in Fragile X and show that we can actually come to a quite definitive decision about rejecting one of those hypotheses in favor of the other through the use of these animal models. So let's talk about Fragile X syndrome. In some sense, pardon the pun, itself is a model disorder. Why is it a model disorder? Well, it turns out to be popular as disorders go. It's an X-linked genetic mutation. It affects 1 in about 4,000 males and about half as many females. Most males have some sort of a mild to moderate intellectual disability. Fewer females have intellectual disability. And importantly, for today, approximately 30, the number sometimes changes, of these Fragile X patients, these individuals have features that would put them on the autism spectrum. What is actually the defect, the genetic defect? It's very, very well described. The normal chromosome, X chromosome, has a region where there are a number of these CGG nucleotide repeats. A typical normal would have up to about 54 of these, and there's a repeat expansion that can occur that causes these repeats to hit more than 200. When that happens, the gene gets silenced, and the cells choose no longer to make the messenger RNA, and so there's no protein. And so that becomes something that's relatively easy to model. So all you have to do is to arrange in a mouse or a fruit fly, something that you have control of the genome, to take the protein product, Fragile X mental retardation protein, as it's painfully been named, and delete either the protein, delete the mechanism for making that protein. And in the case we'll be studying, there's a knockout or a deletion in the gene. The gene is called FMR1, and there'll be a small deletion in that gene so that when the mouse makes that gene product, it doesn't make a functional protein. It'll be knocked out. And you can see here in this Western blot, when you arrange for that, it's not in the homozygous female. It's partially there or partly there in heterozygous females and so on. So it's something that you can actually measure and have a full control over. What does this protein do? Well, it's very well understood that this protein is a negative regulator of translation. What that means is it represses, its presence is actually designed to repress and cause the neurons not to make as many proteins, other proteins, maybe 400 or so, a number in the hundreds, as quickly as it otherwise might. And in particular, this regulation seems to occur near synapses in the dendrites of neurons. That will be important in a moment. And we know that the loss of the protein causes this intellectual abnormalities and the social abnormalities that you can recognize in ASD. This is just a slide or a panel that shows if you measure how much protein gets synthesized in this region of the brain, the hippocampus, there's a lot more being manufactured in the knockout mouse, the Fragile X model. In particular, the proteins are being manufactured here at synapses cartooned like this, and this is what one actually looks like in in cross-section. And you can actually measure some of these proteins as an indicator of postsynaptic density 95, for example. We can actually go and measure, and we can see that there's more of it here in the Fragile X model. This is my favorite kinase, the kinase that's important for storing and the persistent storage of memories. And in these animals, when they're naive, instead of making too much of it, they seem to make too little of it, which is perplexing, but it is a fact. So just because it's a negative regulator of translation of protein synthesis at dendrites, both of these are proteins at not only on the postsynaptic side of the dendrite, but at the synapses, but some are made too much and some are made too little. So the story itself is complicated. Okay, and it is actually very complicated, and we know an enormous amount of the molecular machinery, I won't dwell on that goes with this manipulation. However, that doesn't tell us very much about why manipulations here or these abnormalities lead to some of the consequences that people who suffer with the disease or people who are managing the, the condition have to deal with. One of the things you can measure, though, is that at synapses, and here's a slice through the hippocampus where we can put recording electrodes and stimulating electrodes and stimulate populations of cells that look like this, and we can manipulate the input to this part of the dendritic tree and record a synaptic response, and we can measure how strong that response is. And one of the, despite this protein set of protein changes, in naive animals, the strength of this input here at the synapse seems to be pretty normal. Similarly, if you cause this input to change with high-frequency stimulation, this is a popular way in the laboratory to mimic a massive amount of activity to measure how easy it is to change synapses. They're pretty normal at this synapse. Importantly, I want to point out these animals are naive. They're just taken from their home cage, as these studies are typically done. I'll show you how misleading that can be in a few slides. Another thing that you can observe, however, is if you, in this case, add a drug that's for a group 1 metabotropic glutamate receptor agonist, you can cause a depression that's relatively long-lasting, and you can see in the knockout animals in the Fragile X model that this is actually potentiated. So there's some dysplasticity, as it's called, when the synapses are stimulated, in this case by metabotropic glutamate receptors. You can get these synapses to change too much. And this is a classic study that has actually been fantastic work that led to a general theory of Fragile X syndrome, and including clinical trials and an effort that has turned out not to be as successful as initially planned, but is a really great example of how this work can turn out to be translational. We'll come back to this dysplasticity in a moment. So we've in some sense talked about the genetic or molecular perturbations, how that might lead to different receptor types and signaling, which can lead to synaptic changes. Let's now look at the other side of the divide here, at what we know about the behavior of these mice. So one of the things that you can recognize is in these Fragile X model mice, if you stimulate them, for example, by playing an alarm, they're hypersensitive. They will, if you play an alarm, they will start to run wildly, okay? And in many cases seize, and about 30 or 40% while they're seizing will die just because of the alarm ringing. And you'll see it only takes about a minute or so before they're in this seizure state. So they're hypersensitive. They have some social abnormalities. They're quite normal in preferring a mouse to an inanimate object. But if you change a familiar mouse and pair it with a novel mouse, wild-type animals will prefer the novel mouse. We call this social novelty, and the Fragile X mice will not. They're a chance in whether they spend time with the familiar mouse or the novel mouse. But their memory, if you come back 24 hours later, they'll prefer the novel mouse again. So they have some kinds of social differences, but it's not clear from this whether you would say they have a weird social life or not. Something that I'll focus on here is their cognitive abilities. I'll just start these two movies. So this is a task we'll use quite a bit. We call it the place avoidance task. So there's a mouse. It's on an arena that's rotating at 1 revolution per minute, and its job is not to be detected in what's marked in red. The area marked in red is marked in red for you and me. The mouse can figure out where it is by looking around the room. And if it gets detected in there, the computer will electrify the floor. Not so painful, but unpleasant, and the mouse prefers not to get its feet shocked, so it tries to stay away. And we can measure easily how well the animal has learned the location of that shock by measuring how many times it'll make errors or by entering that shock zone. And you can see that these mice learn quite normally, and they actually remember across 24 hours very well. However, if after this initial training we move the shock zone to a new location, the mice then become impaired. So let's think about what that means. In this condition, the animals now have 2 memories, if you will. They get shocked, and they have the memory for where they were shocked yesterday or previously, and they have a fresh memory for where they were just shocked. And there's nothing that signals where they should go, but they have to make a choice. Okay? What am I going to use to guide my behavior? And these mice tend to be inflexible when they have the FMRP protein missing. They tend to persist in avoiding the initial shock zone and not respond as quickly as a wild-type mouse, which very rapidly abandon avoiding the old area and start avoiding the new current location of shock. I'll explain why that happens by the end of the talk. No, the shock zone is only detectable by being shocked.
And the one fixed object outside.
And they're in a room that they can look around. There are curtains, stuff on the curtains, and so on. Okay, and they can know where they happen to be, and this is important, on the floor. We didn't put anything on the floor, but they put stuff on the floor. And since I'm in polite company, I won't describe what that is. But mice and rats are vermin, and so immediately upon entering a room or a floor, they will mark it, okay, whether or not you can see it. You can certainly smell it if you had the nose. Okay, okay. So these animals, where we've deleted or made the FMRP protein non-functional, where it's been lost, have a variety of abnormalities. We know a lot about the molecular consequences of that, and they have a bunch of symptoms, if you will, that are familiar to people who suffer the disorder. They have a seizure susceptibility, they're hypersensitive, they have these cognitive inflexibility phenotype, and so on. But they have pretty normal looking memory. So the problem here is, how do we go from the molecular description to these behavioral descriptions? So we're going to talk about this by asking this question. And this is now work done by these 3 members of my lab. You can really just pose the question like this. Why does this synaptic dysplasticity, the abnormal synapses I just described that are associated with the protein loss, why does this cause this abnormal cognitive information processing? That's the question we'd really like to answer. And it's very difficult to answer that by studying people, because you cannot measure the kinds of things that are between the synaptic abnormalities and the behavioral manifestation. We don't even know what they are, to be honest. much less be able to measure them. But we can have hypotheses that are quite reasonable based on what we know in neuroscience. So one possibility is because the gene has been missing and proteins have not been trafficked and manufactured for the entire life, including the developmental history of these animals, it might be that their nervous systems are just abnormal in functioning. The neurons that are there that might be tuned and designed, if you will, through evolution to respond in particular ways to stimuli in the world might be abnormal. And that's, in many ways, the sort of common expectation. It may also be that they are fairly normal. Okay? And as spoiler alert, as I told you, hypothesis 2 is what the data is going to recommend. And it may be that their problem is that these normal appearing cells have difficulty interacting with each other. And I'll unpack that in a moment. So this is what the animal models turn out to be enormously valuable for. I don't know if these animals have Fragile X syndrome, but they allow us to test whether the loss of this protein produces neurons that are functionally normal or neurons that are functionally abnormal, and neurons that are functionally normal that have difficulty in controlling how they interact with each other in time. What do I mean by that? And why are we so interested in this timing interaction? So you can frame one of these hypotheses this way. This synaptic dysplasticity that we observed, this is going to disrupt neural information processing and how it's represented in the brain. And you can think about this if we go to this analogy of a traffic light. Each light here is, if you will, analogous to a functional neuronal type. So when this light or that set of neurons becomes activated, it's going to signal something very clear and unambiguous, in this case stop. There'll be different neurons that are dedicated to signaling some other signal, for example, the green light here, go. And what you would expect, what you would predict in this case, is that as shown here with these railway crossings, there's no train here. The bars are still up, but the lights are on. Some sort of dysfunction and disorder in how these lights or these cells themselves function. Here the lights have failed to go on to signal the train. So we would expect, if we knew what signal to look for in the brain, to not find it, if you will, according to this hypothesis. On the other side, you can think about the brain as being a collection of neurons where the important thing that signals information is not so much which neurons are present, but how those neurons interact with each other. Here's a cartoon example of this. A stadium jumbotron. If you're a sports fan, then you stare at this all the time on the weekend. There's a gazillion lights here. Each light, you can imagine, is analogous to a neuron. No particular light or neuron in this example is important for this message. It's not the number of lights that are on. It's not the number of lights that are off. It's not where the lights are. It's their pattern together. And the pattern is defined by which neurons or which cells, which lights in this case, are on and off together. Right? The timing or the coactivity turns out to be important. And so this hypothesis would predict that if we were able to measure it, it'd be the messing up of the spike timing between the neurons that would explain this dysfunction. So how do we proceed? We're going to use this task, and here what I've shown you is after mice learn this task, we can genetically engineer the mice so that they will label the neurons, in this case the hippocampus, that were active during the learning of this task. And you can see there's about 30% or so of the cells in the CA1 region and in the dentate gyrus that have become activated. There's some nice details here that our research is exploring. You'll notice that this ventral blade of the dentate gyrus is much less activated than the dorsal blade. That turns out to be important in our follow-on work. And what we're going to do is we're going to measure the electrical activity of these cells, in particular the CA1 cells. We're going to measure it as the animal walks around the environment, and we're able to measure their action potentials and sped up here. The gray is, imagine a mouse walking around very fast. This has been sped up 100 times. The red dots are where the action potentials were emitted, where the mouse was when the action potentials from one of these neurons is emitted. And you can see that this cell seems to signal something about where the mouse is. Okay? And we call these cells place cells. The discovery of place cells was awarded the Nobel Prize in 2014, in large part because it is a cognitive signal. It's very difficult for you to show the sensor, if you will, for place. You don't happen to have an epithelium like you have a retina or hair cells or something like that that can detect locations in space. But nonetheless, the brain, this part of the brain, the hippocampus, is able somehow to compute information about place, and we're going to use that as our signal. One other thing to make clear is this neuron signals locations, but it's receiving on the different parts of its dendritic tree, in some sense, the components of making locations. It receives a border signal from some cells, a directional signal from other cells, a distance signal from all of this in the entorhinal cortex, the cortical structure that projects to this part of the dendritic tree of this neuron, and it's receiving from CA3, this region, another place signal. And so the CA1 cell is receiving 10-ish thousand of these inputs, and it has to integrate them across its synapses. And remember, we're interested in a mouse that has abnormal synapses, at least defined biochemically. And so let's see what we learn when we record a bunch of these neurons in a mouse that's freely behaving and able to walk around. So the first thing to note is that there's nothing interesting here on this busy slide. This is just characterizing the basic electrophysiological properties of the pyramidal cells that are place cells, as well as the inhibitory interneurons here. They have, whether in the wild type or the Fragile X model, the same features. There's no differences that we can detect. How about the place cells? Well, these cells look absolutely normal when recorded in the Fragile X model or the wild type animal. This was extremely exciting for me, entirely demoralizing and depressing for the folks in my lab. I was very happy about it because I had the ability to reject, even if it was the common hypothesis, one hypothesis in favor of another, so we proceeded. However, a proper skeptic might say, well, are these mice actually in your hands, Andre, at all like the mice that have been described in the literature? And the answer is yes. I'll just show you one slide. We can do the same slice physiology that I showed you before. In this case, we're going to do the physiology in naive animals as well as animals after they've been trained or after they've been exposed, just been allowed to walk around in this rotating arena, never being shocked. They don't have to care about where they are. And when we look there, let's look here at the wild-type animals. So the animals that have been just walking around here in gray or naive, they have an input-output curve which measures the strength of their synapse that is quite unchanged by just walking around. However, if they get this training, that synapse strengthens. If you look at the Fragile X model mouse, this is the naive. The naive looks exactly like the naive in the wild type. However, just walking around the arena creates a massive enhancement of that synapse, and the training creates a yet larger enhancement. So these animals have some abnormal plasticity. They become, with experience, more modified, if you will, even if it's not a memory task. They have that dysplasticity. If we look here, can we elicit changes in these synapses? After training, if you create this high-frequency stimulation in the wild-type animals, what happens is the synapse becomes harder to potentiate. It potentiates, but it doesn't potentiate as much as if the animals were naive or didn't have the memory training. And you get rather a different picture here. In the Fragile X mouse, they happen to have the same pattern, but the strength of the synaptic changes is substantially more. So there's something up with the synapses, But functionally, these cells look quite normal. You can use the different place cells to actually decode where the animal might happen to be. And if we, which is something we'll do later, and I'll show you that that also looks quite normal. So it's quite clear that we can't describe the cells in this mouse as being disrupted in any straightforward way. All of the functional properties that we were able to measure and that people typically measure in such a system turn out to be normal, despite all of these molecular and synaptic abnormalities that are well characterized in our own hands as well as the hands of our colleagues, which allows us in some sense to at least not favor this disruption hypothesis. So expecting that the brains are dysfunctional because of some developmental program that caused the neurons not to be able to respond reasonably to the environment is unlikely to explain the symptoms that one sees. Before I start talking about timing, in particular, I need to talk about timing in general. So here's a section through the hippocampus, and if you put electrodes in these different layers, if you will, of the hippocampus, you can record these local field potentials, these oscillations, and they look different. Even if you move the spacing here is about 1.5 or so, or 150 microns. And so you can recognize that these oscillations differ. These oscillations are the field electrical activity from a massive number of synapses that are being coherently activated. And because of the physical arrangement in the hippocampus, you can measure these oscillations. Why is that going to be important? I need to tell you about my attempts to learn to surf. So I love going to the Society for Neuroscience meeting. I happened to miss it this year. I was very depressed. Why? Because it was in San Diego, and you can surf in San Diego. At least the water's a lot warmer in the fall than it is around here. And I turned out to be a really bad surfer, and so I spent a lot of time in San Diego trying to surf, thinking on the water instead of surfing. And this turns out to be a rather useful thing for understanding why those oscillations were important. I'm going to show you a movie of these surfers. Think of them as neurons who are encountering a wave of synaptic input that's arriving as a population. And let's see what happens to the surfers. Okay? So all of the surfers actually are receiving the wave, if you will, but only 3 of them, the ones that catch the wave at the right phase of the wave, actually capture the energy of the wave. And if we were to watch this, you would actually see that because these 3 surfers were up on the wave, there's a rule in surfing, one dude on a wave, they actually forced and caused 2 other surfers to abandon riding the wave. They were in some sense suppressing that possibility or inhibiting it. And this is actually what turns out to be the case when you put networks of neurons together. Neurons that get the synaptic activation at the right time in this oscillatory pattern are promoted and given a bias, if you will, to become activated. And the ones that are activated early in that sequence tend to suppress the others through interneurons. Okay? And I can show you that we can measure this by this complicated movie, which is meant to show you how I'll make a picture. You can record one of these oscillations. You can filter it for a particular frequency. Here, let's say about 8 hertz. And you can measure where on the oscillation, at which phase of the oscillation particular action potentials happen from a particular cell, and you can plot a histogram of the phase at which these spikes happen, and you can see there's a preferred phase here at about 180 degrees, and the cells tended not to like to fire at the phase 0 at the crossing there. And so you can build a color coded map, if you will, for the preference for spiking at a particular frequency at a particular phase. So if you imagine this histogram turned on its side and the peaks being represented by the hotter colors, you would get that stripe. And you can do that for different frequencies, and when you do that, you end up with a picture that looks like this, which describes the probability that a particular neuron wants to fire at a particular phase, okay, of an oscillation at a particular frequency. And if you look at this, you can see that these are place cells in the two types of mice, as well as non-spatial pyramidal cells and interneurons. They all make these phase frequency maps where the cells have a particular preferred timing of when they like to fire relative to the ongoing synaptic population activity. And we're going to use that. And so what we can see is in the place cells of the wild-type mice in the Fragile X model, there tends to be no particular phase preference. They like to fire at the same phase preference. There's no particular difference. However, there's a strong difference between the rest of the pyramidal cells that are not acting like place cells, which is the majority of cells, as well as the interneurons. And this is happening at this theta frequency along here, about 8 or so hertz, as well as changes in the higher frequencies here at gamma, which I'll talk about in a moment. So you can quantify this. There turns out to be a difference there. And in particular, we'll focus on this. If you take any pair of cells and say, how do two cells like the same phase of firing? Do they like to fire together in the same way relative to the local field potentials or not in the theta range or the gamma range? It turns out that the animals in the Fragile X model happen to not like to be like each other. They're not as coordinated. The taller the bar here, the less coordinated they are if they're place cells. Okay. However, the interneurons and the non-spatial cells are massively different. They are very stereotyped in when they like to fire. They create, in some sense, a rigid framework within which these cells have to sit. And there's very much lower variability in how they will discharge relative to the ongoing spikes. If you look at pairs of cells and just say, hey, here are two cells. Imagine you pick two lights of that jumbotron and asked, how similar do those two lights flash or blink together? That's what we're measuring here with this factor tau. And what's shown here is the first and second half of a recording. And what we've done for each cell pair, we've organized the cell pairs from 1 to 247 in this case, and we're showing the correlation of that cell pair in the first and second half. And you'll notice in the wild-type animals, they tend to fire quite reliably from the first half of the recording to the second half of the recording, not so much in the Fragile X model mice. So these cells, if you imagine them representing a jumbotron, in the wild-type animals are very reliable in their interrelationships, but they're not so reliable. They don't interact in a reliable way in the Fragile X mutant animal. After training, what we see that happens is that there's this plasticity that comes into play. And what we can observe if you look at this histogram at the thick line, this is a description of that correlation and the likelihood of observing a particular correlation in the population. And before compared to after, this experience doesn't change very much the distribution of these correlations in the wild-type animals, but in the Fragile X model mice, you get a big rightward shift, meaning that these correlations became very positive. So which is the kind of thing you would expect if the network became very deterministic, if you will, of how the cells should fire together. However, it's not the same cells from one moment to the next. It seems to be, that's what I showed you before, it seems to be that the whole network gets plasticity, but it's spurious plasticity. The same cell pair might like to fire together early in the trial, but not late in the trial, and vice versa. So if you think about interactions amongst pairs of elements like I do, you might really appreciate these mirror mutation movies. You can see this also on the Foundation website. These are absolutely fascinating things to see. These, tens of thousands of starlings flying around. You can see the same phenomena in fish that are schooling, and they will make these complicated patterns from very simple interaction rules. It turns out that these very complex arrangements of flocks happen almost instantaneously, and the rules that describe what's going on are a particular starling is sensitive, if you will, to the 7 nearest neighbor starlings. And we call this scale-free correlations across the whole flock. And it's these kinds of interactions that I've just described for you that seem to be abnormal in the Fragile X mouse. So that's precisely what was predicted by the discoordination hypothesis. We didn't pull that out of thin air. We had seen the same kinds of phenomena in other models that maybe represent things like depression or schizophrenia in particular. So that's not really helping us across the gap. We've gone a little bit further. We've identified an interaction, if you will, that we might be interested in describing. Let's go to the last part of the talk and try and explain why it is that these particular mice have difficulty when they're confronted with a new situation that conflicts with what they had initially learned. Excuse me. So this is the story, if you will. What I'm going to describe now is a bunch of place cells, right? Cartooned here is 3, that are able to discharge. And the discharge is going to be exquisitely timed relative to this ongoing local field potential, not only at the theta or 8 hertz time scale here, but also at this very fast or faster gamma time scale in the range of 30, in this case 30 hertz or 70 hertz here. And what we're going to see is that when these cells discharge, typically they discharge to signal where the animal turns out to be. That's the definition of these firing fields. They're place cells. However, there are moments when in this oscillation we see that the slow gamma here, this 30 hertz signal, is dominant in the local field potential. And when that is dominant, these cells no longer signal where the animal is, but signal someplace remote from the animal, some distant place. And in particular, what we'll do at the very end is recognize that a wild-type mouse, given this conflict between a familiar location of shock and a new location of shock, when it experiences these moments here, we'll talk about them as being recollective moments, it will represent the new location of the shock, whereas the animals that are the Fragile X model, they will also show a remote encoding. However, they're going to pick the original location of shock to represent rather than the current location of shock, which is going to explain why they're inflexible in their behavior. So let me show you the evidence to make that story. So what we can do is put the animal on this rotating arena, and we can not feed the mouse. That's why there's a grid floor, so there's nothing interesting in that arena. And mostly the mice will just sit there and get wheeled closer to the place that they should avoid. And when they get close enough, they'll turn and walk away and run away. And sometimes they fail to do that and they get shocked. And if you measured the distance to the shock zone, you can see that it has this sawtooth pattern where the green stripes are the moments where the mouse is just sitting there and it's being wheeled closer to the shock zone. And for some reason, it's going to run away. Okay? And you can see this ballistic movement. So these green moments are very interesting for us. The mouse isn't doing anything. The mouse is in the same state, if you will, across all the green moments, no matter where the mouse happens to be. Okay? And it's going to run away. And why would it run away? Okay. Presumably it's running away because it is remembering, oh, I'm near the place where I'm going to get shocked. That was a guess that we made. And so we were very interested in looking at what happened in the brain, in the hippocampus, moments before the animal ran away, guessing that the animal must be remembering that it had been shocked in the past. And this shows what we observed. So we can record the local field potential in the pyramidal cell layer of the hippocampus of those CA1 cells that were place cells. And what's shown here in this time frequency plot, where the hotter the color is the strength of the oscillation at a particular frequency here, you can see that at time 0, which is when the animal is going to run away, for a couple of seconds before that, there's not that much of this medium frequency gamma in the 70, 80 hertz range. You can see a lot of it after the animal runs away. And there's a lot more of this slow gamma activity, this slow oscillatory activity here in the 30, 40 hertz range for a couple of seconds before. And when the animal is successful, you see more of this slow gamma compared to this medium frequency gamma. And when the animal makes an error, you can take the ratio of these two and you get this plot. When the animal makes an error, you don't see this change. So that's making us feel comfortable, at least, that this has something to do with the animal being able to escape. And in fact, you can use this ratio of the slow gamma over the medium gamma. When that is high, we call it slow gamma dominance. The animal, you're able to predict quite well that the animal, at least above chance, that the animal is going to actually make an avoidance in the next 3 to 4 seconds. Okay, so we might have a signal that has something to do with the animal wanting to get up and walk away. So there's another thing that we can do. We've got our electrodes in the brain, and we're able to record sets of these cells. Some of them are place cells, some of them are not place cells, some of them fire sort of more or less in a messy way. But we have patterns of this activity, and if you imagine this is something like that jumbotron, we can look at the particular patterns that we're able to record. And we can do that instead of averaging across tens of minutes, as in this case. We can look at these moments during short moments of time. We're going to pick 400 milliseconds, so a fraction of a second, and say, hey, we've recorded 53 of these cells. What is the pattern of activity analogous to what's the snapshot of the jumbotron across this short moment of time? That's the state, if you will, of the system. And we can do some math with that and ask, how likely is that short pattern of activity able to tell me where the animal is? And so what you're going to see are these formally Bayesian posteriors, a plot that looks like a firing field from a cell, but it's actually what the whole ensemble that we're recording is pointing to, the probability of where the animal might be. And in this case, this observation says, hey, the animal is likely to be in this area that's colored with the hot colors, unlikely to be where it's dark blue, and the animal happened to actually be where the X is. So we can actually decode quite well, it's sort of a mind-reading trick, how well the hippocampus is signaling or representing where the animal happens to be. And what we notice, let's just focus on here, we can look at this ratio of the slow gamma to the medium frequency gamma here in the red line. And when that is peaked, what we notice here is that the probability of where the animal is turns out to be distant from where the animal actually is. And when this ratio is low, we actually turn out to decode accurately. So the activity, this is in a normal mouse, is changing as the animal walks around. Sometimes it's signaling where it happens to be, and at other times it's signaling some other location in the space. What is that other location in the space? In this case, we can't tell because we just have the animal walking around, but we can do an experiment. we can have the animal doing the place avoidance task. And I'll show you in one, what is this, 12-second sequence, what we can observe. So at time 0, the animal is going to walk away from the shock zone. The arena is rotating in a clockwise direction. 6 seconds before, we're decoding local to where the animal is. 4 seconds before, decoding local to where he is. 2 seconds before, the decoding starts to point not to where the animal is, but where he's actually not going to go, as well as where he is going to end up. At time 0, the animal is going to turn and walk away and come over here. Again, at time 0, the activity in the hippocampus is signaling not where he's going to go, but where he's not going to go and where he's going to go. Afterwards, the activity starts to catch up with the animal and starts to decode again locally. Okay. So it's very fast. You can't see this if you average across the 10 minutes in those maps. Here's another example of that. In the interest of time, I'll just show you what happens when the animal makes a mistake. 6 seconds before, it's decoding locally. He's going to enter the shock zone and run away here. It's still decoding locally, always local. Okay? When the animal makes an error, the hippocampus continually references where he happens to be, rather than making this remote, if you will, representation that's transient, only lasting half a second or so. What happens if you now forget where the animal is, but just look at the likelihood, what this probability map looks like on average whenever you find slow gamma dominating medium gamma? I haven't told you, just in the interest of time, but please ask me, because I'd love to explain why it's important that slow gamma dominates medium gamma. There's actually a very explicit biophysical reason for that being recollection. But what we observe then is when slow gamma dominates is that no matter where the animal happens to be, he might be here, 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 anywhere here, whenever that occurs, the probability map of what the hippocampus is representing is where he's not going to go. You can see this matches very nicely the region that he isn't going to go around the shock zone. And if we pick the same number of random times, this is the picture that you would get. So these slow gamma dominant moments seem to represent not where the animal is, and not, in this case, not randomly some other place, but in particular the place, if you will, that the animal must be recollecting about where it had been shocked, because it's not going to go there. Remember now, these Fragile X knockout mice, or Fragile X model mice, have quite normal learning and memory, as measured here, when they happen to learn to avoid one shock zone. But when you change the shock zone in these conflict sessions, then they show you this inflexibility impairment. And if you now look at those slow gamma dominance events as shown here, they're more or less normal in the rate, okay, during the learning and memory test. But on the conflict test, the animals that are wild type suppress this. Whereas the animals, the Fragile X model animals, if anything, increase this, but they certainly are unable to regulate this. I won't tell you the mechanism for this regulation. It's something that we're actively working on in the laboratory, but there's a synchronous event from the entorhinal cortex that is regulating that, and is something that we can actually target and influence. And this is the last data slide. So this is just another example of what happens in the wild type and the knockout mouse when the animals encounter the shock zone and decide to walk away. This is what I showed you before. But what happens when the same thing occurs, same situation, but the shock zone has been moved? Let's look at the wild type mouse here, right? It's decoding locally, right? Locally. Changes to remotely, and the remote decoding now aims at the new location of the shock zone, which it will then avoid. Okay. The Fragile X model mouse, however, is encoding locally. Okay, locally, just before it decides to run away, it decides to start encoding, if you will, distant from where it happens to be, but it's not aiming at the shock zone. It's aiming at the previous shock zone. It's using its memory. In some sense, it's stuck in this slow gamma-dominated recollection mode. And that's what this mess at the bottom is quantifying. So I'm hearing all these bells, which tells me I have to finish, which is what I'm going to do. I have a minute. So to summarize, I hope I've convinced those of you who don't care about fMR1 mice or place cells or oscillations and so on, at least that using animal models is a really powerful way to test ideas about the human condition, so that you can actually get a mechanistic understanding of this complexity. I hope I've convinced you that the loss of this protein causes some kind of cognitive inflexibility, and we actually know why that happens. It's because of this discoordinated timing of the neural discharge amongst a whole bunch of neurons, and it's not because these neurons themselves individually seem to have dysfunctional properties. This hypothesis, the discoordination hypothesis, right, that asserts it's the discoordination that underlies this inflexibility, offers a rationale, a way to think about this problem. It offers, because we can think about it, an experimental platform. I've shown you the experiments and the kinds of experiments that we can do. And in fact, what's interesting is it actually identifies that these coordinating properties is something that we can actually target and try to influence. We can stimulate the brain, and we can use various kinds of modulators to actually manipulate that ability of cells to fire together and not fire together. And finally, one of the things that we're trying in an open or closed loop system, you can stimulate and do what would be called neuromodulatory tricks, the kinds of things that you would recognize with a cardiac pacemaker. And it's these manipulations that show us that we might be able to effectively disrupt this abnormal coordination. When the brain is stuck in the recollective mode, when it should be confronting and collecting new information, we can stop that by this kind of stimulation, which might provide at least an interesting therapeutic target. So with that, I'd like to thank all the people who worked on this project. In particular, they are highlighted in red, and the funding sources, and in particular, the Simons Foundation that started all of this work by a pilot grant. So thank you for your attention.